Okay, there is nothing important there. That just so to just set the scene a little bit, I can, yeah. So I will just set a little bit the scene of how I am thinking about my questions. So in the ultimate end, we're interested in this very complex system, and in my understanding, we can decompose this complexity into studying on one side a complexity and features of the structure. On the other side, we have the intricate dynamics. Here is in dynamics of a very, very simplified nervous experimental system of dissociated culture, but this shows a lot of complexities that might be interesting to study. And to make a full story of it, we also need to incorporate what this system actually doing. So it tasks, its goals, its behavior. And my vision is that we need to unite these objects to understand brain states. It's something that I might tell you a little bit about. And this simplification would possibly allow us to understand a bit better where and how the brain self-organizes. And if we would be able to define something that is capturing together the structure, the dynamics, and maybe a behavior, then we would be able to use this simplification to make sense of this complex dynamics, and possibly then also to use it for diagnostic and treatment. So what do I mean when I say brain states? There are some brain states that we can very easily label behaviorally. So, for example, we can label different sleep stages. This is something we understand very well in behavior. We even know how this corresponds to changes in dynamics of different brain areas. There are some other states that are somewhat behavioral, but typically seen only in a statistical sense. Like after many repetitions, we could distinguish attention versus non-attention condition, or we could measure the pupil dilation, but we also need to do it on the multiple trials. So it is hard to spot on to really understand on the spot whether Damien is paying attention or not.
Split attention between the waiting and
I am just trolling you, I'm sorry. So, and there are some types of states that can be observed in activity, but we don't know how to map them to the behavior yet. And there is no strict definition of how to actually merge all of the things together. So now just few words about states that we can see in activity. The simplest ones are related to synchronization. As I briefly showed in the sculptures, we have a very clear synchronization patterns. On the other side, we have even much more studied phenomena of looking at the synchronization in hippocampus in form of sharp wave ripples, which is known to be useful for memory consolidation, but we don't really map it very clearly one-to-one to behavior. And we can see it only in the recordings of activity. The other type of states, which I'm personally very interested in, are marked by closeness to so-called critical points. And here's just a small cartoonish way of how I sometimes explain what critical point would be. So imagine we have a system where we can put some information in and look how it spreads, for example, in this room or in the whole society. And sometimes it does not spread at all, for example, because it is classified. And on the other side, it might be that Elon Musk tweeted something stupid, and so everybody knows about it next time. It takes over the whole system. But what we are typically interested in is this part in the middle, which has a certain chance of spreading, but typically remains small. And it is mathematically very well-defined point where every information spreading step is barely possible. So on average, each person would spread information to single other person in the network. And here we can see such spreading event in the 1D lattice. This is the illustration from very nice review by Miguel Muñoz. And here we see, so this is a lattice itself, so it's just one row of neurons, and this is time. And here we have a subcritical regime where information not spreading on average. So it would at some point maybe spread in the beginning, but stop. On the other side, we have this information that takes over the whole network. So in the end, everybody knowing it. And in between, we have this complex structure, which can either spread as this beautiful tree, or it could also die out. And it is typically characterized by the distribution of how many people, in this case, would be informed. So today, I would be a lot talking about a simple proposal of looking at the timescales. So before I was showing you the spatial spread, but we could also look about it on it at time and using this timescales of activity as a proxy for the state. So why the timescales are interesting? One of the works that kind of started the big interest in intrinsic timescales it was a work by John Murray, who looked at the intrinsic timescales. Intrinsic meaning that you remove the timescale that are coming from the input. Here, a monkey was not doing anything specific, and the activity was recorded by multiple electrodes in the different brain areas. And by a particular method John used, it was possible to measure the timescales. And what they found that along the anatomical hierarchy, there is also a hierarchy of timescales. So, for example, if we would look at the we don't see them here, but if you extend it to the V1, like primary visual areas, they would have the shortest timescales, and then it would go longer to more, to higher areas that are requiring more complicated computations. And this way, this was very logical. So the area that needs to compute complex things, need to relate signals across different times, need to have a longer time scale. So it needs to forget slower the signals that were presented before. So this was very exciting for many people who kind of tried to make sense of the brain in total. And afterwards, it was also shown in the different modalities of recordings. So the first spiking activity in monkeys of John Murray, there are some people who try to see whether the same thing is working with mice. There are ECOG and MRI showing the same type of hierarchy. Yeah, the question is, do we actually know how to measure them nicely? And are these measurements from different modalities showing the same thing? On the other side, like people measure them somehow, and then afterwards look, are they meaningful not only as a hierarchical thing that is just there, but for performance on a particular task. And it was shown that, for example, if you look at the neurons within a certain task, different timescales of neurons with different timescales would fire in the different precisely selected parts of this task. So they have something, they code differently. And then another part, it was shown that if there is a certain task a subject performed, here is a human subject, then after performing this memory related task, the timescales would be longer. Because if you need to memorize, you possibly need longer timescales. So this might be a bit speculative, but the non-speculative part is that after performing the task, the same way measured timescales were longer. So here, a little bit of a structure, what I am going to talk about in the next moments. One part is we had the timescales that are the whole brain areas, or maybe individual neurons. I will talk about neural populations. So the question is, are there some different populations that would have different timescales, and do they have any specific roles? Then I will talk about things which I like a lot, is modeling, which mechanisms could contribute to these timescales. And this is work done primarily by my PhD student Roxana, together with a group at that time at Cold Harbor Laboratory of Tatiana Engel and her postdoc Yang Lang. To do this properly, it appears that we actually need a very good method to measure the timescales. And so that's what we spend a lot of time developing. And then if we have time, I would also tell you a little bit about how the diversity might be used in a very simple computational model. Okay, so let's start. First of all, let's define mathematically what we typically mean when we talk about a timescale. So when we talk about timescale, we often think about the autocorrelation function. So here we have, for example, a bint spike train. And for a 0 is there is no spike within this bin, 1 there is a spike. What we're doing, we are shifting the time series by a certain time, and then looking how similar are they in terms of correlation between the time series and the shifted time series. This can be measured as a function of how far we shifted it. And this is an autocorrelation function. It takes as an argument the shift and gives you how related are the time series after you shift them. This is what you typically see if you would plot this function. It typically has an exponential decay. So if we want to have something with exponential decay, or we want to check whether something has an exponential decay, the best thing is we plot it in log linear scale for and then we have this very nice straight line, and the slope of this line is a time scale. It tells us how fast this exponential decays. If tau is a very small number, it means that it decays very fast. There is no connection between the time series and the shifted version after very short time. And if tau is very large, then this is almost 0 for a very long time. So this part would decay very slowly, and there would be still connection between the information in one moment of time and information very long time later. So imagine we could measure this tau directly. Now let's go to see the data. We use the data that was already recorded for a different task in a monkey V4 by this multi-electrode array that it inserted perpendicular to the cortex surface. So basically all electrodes are recording the same receptive field. And what we did here, we compressed all the slides for all the spikes from all the 16 channels. These are putative spikes, and it is fine to do so because they are very synchronous, as Tatiana showed in her previous work. The major changes are happening simultaneously on different channels. The task the monkey had to do the first task was just concentrate in the center, do nothing. And we recorded, there was short periods of time when the monkey was doing this, and we recorded this autocorrelation function during this time. It's plotted now in this log linear scale. If we squint a bit, we can say that it is straight line. If we squint a bit more, we would maybe think also that here is something different than what is happening here. The other part was when the monkey actually had to perform a task. The task was split, it was to detect the changes in one of these Gabo-like structures. And before performing the task, the monkey was given attentional cue. And sometimes this attentional cue was pointing towards the receptive field of the neurons we record. And the monkey had to report by doing an anti-saccade. So saccade to the other location. This way, these two parts are kind of related to attention. We cannot separate them. So this is attention condition. And the other two are not related whatsoever. And they would be attention away from the receptive field conditions. So now we could split the trials into the groups of attention towards and attention away the receptive field. And this is what we get if we would plot the autocorrelation functions for these two cases. So if we would just look at it, the hypothesis we might formulate just visually would be, first of all, it looks like there is something happening fast, like it decays fast for the small x, and something that's happening slower. So there might be two major timescales that are explaining the activity in this V4. And then when we look here, there is nothing changing in the beginning. They are very similar. The fast timescale seems to be the same, but this one decays slower. So there might be that the slow timescale increases when the monkey tends towards the receptive field in this local population. To test it, we need some kind of like good statistical methods. So that's what we turn to do. And to understand why we need a good statistical methods, I would have to present you what are the problems with measuring timescales the way we're doing it. And to make it very visual, we simulated a classical process, the stochastic process that produces a single timescale data. It is an Orsa-Nulenbeck process, but it doesn't matter. It's just a simplest process that produces a single timescale data. And we know analytically what is this timescale. If we simulate it infinitely long, we would get a straight line with the slope we put in. And now let's do it more like in neuroscience and have some short trials. In this case, it is 4 seconds trials, which are very long for monkeys in the same state. And we see that if we would just feed the straight line to the data that we recorded over 4 seconds of the same process, we would find a different time scale. And if we go to the place where we actually, like our data was only 1 second before stimulus presentation, 1 second after stimulus presentation. So it was kind of this order of magnitude. We would the seconds instead of 80 that we actually put into it. And this is this classical case where this is a problem that was known since 50s. And for some very specific processes, it is solved, but only for very clear mathematically defined autoregressive processes, which is not what we have to use for neuroscience. And what we know that this change in the inferred timescale definitely depends on the duration of trial, depends on the spiking rate, both on this mean and the variance, and depend on the timescale itself. So it is very hard to find a bias that depends on the value. We need to debias it somehow. To do so, we used a simulation-based inference. This is a procedure that allows to find in a Bayesian way the distribution of parameters that give rise to the data without knowing the explicit likelihoods. So what are we doing? We have our measured data that's here. We have the autocorrelation that we computed from this amount of data, this amount of trials, these durations. Now we say, okay, if we want to say that this is a data that has a single time scale, there should be one single time scale data generating process that would be very similar to our data. And we say, okay, we take the generative model, this Orsa-Nulenbeck process, and we give it different values of timescales. And we maybe take multiple of this Orsa-Nulenbeck process because we want to have one time scale here, one time scale there, and then we mix them with a certain mixing coefficients. From this process, we can produce our simulated data. And then we can, what we can do then, we can also control that we produce a simulation data of the same lengths. We can control the mean and the variance. So what we say that we control all the major factors that is influencing how biased the timescale inference is. And then we look at the autocorrelation function that is produced by this process. And there is some distance. In our case, we just measure the integral, how much these curves are differ. And if this distance is small, whatever way we define small, we say, okay, these parameters were actually quite good, and we remember them. We store them as a part of our posterior distribution. In general, what we would be doing, we would be sampling many of these parameters. We would use some additional heuristic to make it very efficient. But the idea is that we are looking at multiple parameters, checking, do they produce a similar data under the constraint that the bias generating terms are the same as in the real data. And then we have the distribution of parameters that would be generating very similar data to our recorded part. This way, then afterwards, we could say, oh, these are the timescales that are likely to produce our data. So let's have a look how this works in the data. But before, if you would want to, we have a Python package that does it automatically, also checks whether this is necessary or not for your data because of the size of the data and so on. So first of all, we tried it on the synthetic data. We could do it on the data that I showed you before, where we know that there is a single time scale, and we know that because it is short, we don't see it. Like here, it is a single time scale that is 20 milliseconds, and if we would just feed it directly, we would get 17. It depends whether this is for us a big deal or not a big deal. And the posterior distribution, so the likely parameters are very close to the real ones. Or we could have an oscillation on top of exponential decay, a very typical situation, and then we would be able to recover the time scale of the exponential decay, and actually we could simultaneously recover what are the oscillation frequencies. Or as here, where we have two time scales, and on top of it, we also have spikes generated. So we kind of produce the rate function and then generate spikes, and we can mimic it and find the true parameters very closely with the Bayesian procedure. Additional advantage of having a Bayesian procedure that we can check, we can test hypotheses. Typically, you can fit a model with one time scale, you can fit the model with two time scales, you can fit the model with 50 time scales. How do you select at which number of time scales you stop? Here, for example, this is the real data, and we fit it with two models. Okay, the model with one time scale looks a little bit worse, but is it really a big deal? We have one parameter, even two parameters less. To rigorously in statistical terms decide, we can use the base factors, and from the posterior distributions and distributions of error, we have them automatically. And in this particular case, what we see is that two time scales model have a very small error. So the cumulative distribution functions of errors grows very fast. And so the errors are always smaller. If the CDF is smaller for the larger for the one model than for the other, you can easily select it. There are less strict criteria, but we were fine enough with the strictest possible. So we only say that in this case, two time scale model wins over one time scale model, and like as it is here. And for our data, we have three different monkeys in two different conditions. The dark green are the ones where it is a clear preference for two time scale models, and the black ones are the ones where we cannot statistically decide which one is better. And in almost all trials, it is two time scale that fit the data the best. So now we can go and fit this way the different attention conditions. And if we look at the posterior distributions for the both timescales, what we see is that in both attention conditions, this fast timescale that captures this part is on, like this is one session, are completely overlapping. However, the slow timescale is very significantly different between these two cases, and it is much longer when the monkey attends towards the receptive field. So this is one session. Now we can pull the data across all sessions, and this remains the same across all sessions and all monkeys, and it remains the same. And what we were very excited about, great question from referees, was to find that actually this is related to also behavioral variables. What we look at here is the reaction time of the monkey. After it's seen the change, it needs to react with the anti-saccade as a function of time scale, of the second time scale, or plotted together with the second time scale. And what we see is that when the second time scale is longer, a monkey reacts faster. And it might mean that it's just kind of integrating better, or is it just a different, somewhat different state when it is ready to react much faster? We cannot prove that it is causal in this setting. However, the data from which we measure the time scale preceding the part where the monkey has to make a decision. So there are indications that, at least in temporal order, it might be actually causal, but we cannot disrupt this without any further ado. And if the monkey was not attending, then the time scale is absolutely not informative about the behavioral measurements.
Sorry?
Yeah.
A block is a unit? Or is it
So,
unit or a different trial?
No, but it's different sessions.
Sessions.
To measure them, we need some time.
Yeah.
But we need some number of trials.
So you suppose that every unit the population is the same thing.
So we unite all the electrodes. We even don't, like, we have few units that were separated, but for one unit we don't have that many spikes. For this method, basically nothing would be significant. There were just very few units where it was enough spikes and we could do the same thing, but this were, I think, like 5% of all units that we had. Okay, so we see that there is something happening. The question is how? And this is where the modeling enters the game. How do we model the processes or networks of neurons that produces long time scales? In the classical balanced network, where we have excitatory inhibitory neurons, the excitation inhibition are well balanced, what is known, the timescales are very fast. You excite something, it's just very rapidly balancing out. There is no long time scales. It was this way until this paper of Levina and Doran, who showed that if we would have clustered connectivity, we can produce a longer time scales. And on the other side, what people were playing with are a little bit more phenomenological neural units that would have different timescales. They would be recurrently connected, but would have different timescales. Some would be fast, some would be slow, and this also can generate a diversity of timescales. So what can be used for modeling? We really like recurrent networks. So this is our very simple model. Every node is kind of this local population. And it could be either active when the state is 1 or not active when it is 0. And the transitions are probabilistic. So if one node is active, it can activate its neighbors. And in this particular case, the neighbors are just the nearest neighbors, for example, but we could spread them out. And the other part is that it can also maintain, because it is a local population and there is a strong recurrent connection between the layers, it can maintain being active. So it has a self-connection. And additionally, it has some input that we modeled in the situation where there is no additional input by just having a probability of getting spontaneously active. If we just use this model with nearest neighbors interaction and record the same way activity here, because it is a model, we can record it for a very long time. We don't want to deal with problems of the short recordings. We see that there is something happening up here and something different happening in the tail. And because it is a model, we know what we put in. One thing that we put in is how it is self-exciting. So if we don't have anything else, but have only this one neuron, how long it would try to keep on going. And this would be its neuron timescale. And this is here. So we see that actually it captures quite well what is happening in the beginning of distribution. The other part we could also find analytically, but we could look at it as what's happening from the neighbors. If we take this neuron and just record everything it gets from the neighbors, then it would get this slow time scale that is mimicking relatively closely what is the slow time scale of this neuron. And afterwards, Jan Lang also did a very cool computations for this model. Now what we can do, we can change. Like we've seen that these neighbors are important for the slow part. What if we would start spreading them apart? Look here, we could use the same number of neighbors, but spread them a bit out. And what would happen, we would see much more stronger and prominently the self connection, and the interactions part would become less important and less important. And then when we just connect them randomly, we don't see the slow part anymore. So this was very exciting for us. And then the hypothesis was that it comes from a clustering connectivity, the slow part. And if we would look at all the neurons together, we would see the same thing, no matter how we spread them apart. But then there are multiple other mechanisms that could generate these two time scales. And the simplest one is maybe not what we proposed. This is already very hard with recurrency. Simplest one would be that there are different types of neurons. For example, exciter inhibitory, and some are fast and some are slow. And when we record them together, we just mix the signals from fast and slow populations, and that's how we can get this signal. Here we just would have one fast population with this time scale and a slow population with the other time scale. And the connectivity could be whatever. The other option that Tetran Storage was working on would be to have slow two biophysical neuron-specific timescales. One would be, for example, its membrane timescale, and another would be much slow synaptic filtering. This also can generate the two timescale autocorrelation if we connect them randomly. And this is our suggestion that all neurons are same, but we have the structured connectivity that generates the slow time scale. It looks like we would have actually hard time distinguishing between them, but they do differ very strongly in how the spatial connections between neurons look like. So if we would measure the cross correlations, we would take the signal in one place, like somewhere here, and would look at how it is connected to the time-shifted version of the signal somewhere else. For these randomly connected networks, first of all, there is almost no connections between neurons. They don't change with distance. And there is nothing happening. In our case, there is a spatial structure. So one thing, what we see is that if we would not take a time-shifted version, we would just go to smaller and smaller correlations if we go away from the neuron. On a big distance, there is no correlation. Close by, they are connected maybe even, so there is a strong correlation. Now the question is, do we have any data to check whether it looks more like this or more like what we would predict? And we were lucky. There is data from some, because in some sessions, they on purpose inserted electrodes slightly tilted. And here, what we did, we measured for all the contacts of the electrodes, where are the receptive fields? And we measured the distance between electrodes, putative distance between electrodes, as the distance between the receptive fields. Because if they look at the different part, they are probably recording from a different population. And here we took, because the electrode was not tilted very strongly, so we don't have very big distances, but we have, we could split very short from very long, from longish distances. And we see, first of all, that there is a structure, and it definitely looks more like this than like any of this part. So this allowed us to convince ourselves and the referees that the spatial connectivity could be a good explanation. Clear enough, it could be a mixture. It could also be we know that there are different types, but at least we need the spatial part to have the structured spatial correlations. And the other part was we could also find the parameters for the models that fits better the attention away versus attention in condition. And here, this is one of the reasons we use this simple model. In this simple model, we know where is a critical line that's corresponding to what I was talking to you before with the spreading. And what we see is that these are all the points that could potentially explain the attention away condition, and these are points that explain attention towards condition, and they are shifted closer to the critical point when as a monkey attended. And this could be change in the regime of the neurons of the local populations, and they are studied in a different context. Here, more temporal or more spatial. It was shown that it is something reasonable for neurons potentially to do. So this would be this first part. I showed you a method how to find timescales from the trial data in an unbiased, rigorous Bayesian way. I showed that the timescales of the V4 local population activity has these two very characteristic fast and slow components. And the slow component, the slow timescale, is changing, is increasing when the monkey attending towards the receptive field of recorded neurons. And this shift, how far it shifts, also seems to be reflected in how fast the monkey would behaviorally react to stimuli. And it looks like this two timescales, and especially the slow timescales that is most interesting for us, is produced by the local connectivity, and that attention shifts it closer to the critical state. So these are these two papers, and then the question is whether you want another 10 minutes of the other story that is not yet published and don't have any.
I think so. You're in time.
So this is a not yet finished story. So the question is it somewhat useful to have the different distances to criticality that are manifested in different types? And now I'll introduce you what we are measuring to understand in the model what could be a benefit of being close to critical point. And so here I introduce you the dynamic range. Imagine this is a schematic plot of what's happening potentially with your response or how you behavior react to hearing sounds. If there is a very quiet whispering on the other side of the hall, the guard is buying a coffee, most probably changes like modulation of this quiet whisper would not really elicit any behavioral response and would not probably be reflected in the neural response to the signal. If the ambulance is entering just in the middle of our room, but it is kind of here or there, small differences in how loud it is, we would probably also not perceive it, would be in saturation regime of our hearing system. So we also don't expect a big changes in neural activity. However, in between, we can very clearly discriminate whether sound is getting a bit louder or a bit quieter. It's for us very important because this would tell us whether a prey or predator is approaching or moving away from us. So this regime where we can, from activity, understand how strong are the signals would be the dynamic range, the range where we can very nicely discriminate signals both in input and in our behavioral or neural output. And we can introduce rigid measures how to measure the size of this dynamic range. For example, we can, as Mauro Capelli and Asami Canucci proposed, we could cut, for example, 10% of lowest and strongest responses, as the responses that are very close to saturation either not responding or responding completely, and then measure. And as with the dynamic range, it is typically measured on logarithmic scale, coming from the Webel of our perception. We can measure this interval that corresponds to the inputs that are within this 90 to 10% interval of responses. And what was shown already some time ago is that if we have this system that can be tuned to different distances to criticality, at the critical point, it would have the largest dynamic range. So it can discriminate between the most number of signals. And this was also repeated in experiments on cortical slices, where they also stimulated and looked at how many signals they can discriminate for this dynamic range option. What we ventured to look at is the following, together with Viola Prisman, her postdoc, Johannes Tinnebeck, and Sahel, who was doing the master. This whole story of dynamic range was made from assumptions that the system can hear for infinite amount of time to the signal and produce a single variance-free input-output relationship. However, in a realistic situation, we have a finite amount of time. So we could listen to this part, or maybe we could listen to this part or this part, and responses could be a bit different for the different parts. So there would be a variance in the responses that is generated by the finite observation lengths. And now let's imagine that we have the strongly subcritical network. We push a signal, the network actually by its own dynamics does not really do much. So here are two different signals that are producing different responses. And this is a long time series. And if we would look at the 1 second bits of it, they all look approximately the same. And we can very clearly discriminate a bit quieter signal from a bit louder signal. If we would take the same level of responses, like here we'll see this in the response curve, there are distributions which we could analytically measure for these responses. And now let's take a little bit closer to critical system. Closer to critical system would produce a lot of activity by itself. So in this particular example, for the same mean responses over 1 second of time, they would have a huge variance of what we observe. Here, the response curve would look very different, and these are the distribution of responses. Although means are the same, you can hardly say that from 1 second of observation, you could really say, was it a loud or a quieter signal? So we introduce a measure for how good the system actually can, like, not in this idealistic infinite observation time, can perform the dynamic range discrimination. Namely, we could look at the stimuli and check how much they are discriminatable by looking at the overlap of the distributions. So this part is where the system would make a mistake. It would not know which signal is presented. And now we could fix this error. We could say, okay, I want it is not mistaken by more than 5%. And now we can spread out signals such that they have this 5% overlap. So we can then compute how many or how far apart they are coming if we are allowing only this amount of error. And this would be accounting both for the variability in the signal and variability that is generated by finite observation times. So now we would have these different versions of defining dynamic range that actually works with the different observation windows. And what we see, if we just look at the classical dynamic range and we approach critical point, here I just show the below criticality and approaching it. This is an infinite time. This is the same curve we've seen before. It's just that I am sampling it much finer. So it is getting larger towards the critical point. But if you take a smaller observation windows, there would be an optimal distance to criticality that is corresponding to this observation window that is canceling out the amount of noise that we have due to the short observation. And this is what we see here. Basically, for the different ways we would measure the the dynamic range, either number of discriminated points or the range, or maybe a mutual information between input and output, we would have the optimal state of the network depending on the time windows. It would converge to the critical point if the time goes to infinity, but it would be always subcritical if it is not. And here it would also depend on the fire rate and multiple other parameters. But what we see is that for short windows, we prefer to be in a more subcritical state. And for longer observation windows, in this case, where the noise is not changing, we would need closer to critical systems that would deliver an optimal dynamic range. From this, the assumption that maybe what's happening under tension, we know that there is some of the noise suppressed in a tension condition. So if the noise is suppressed, it might be better to move closer to the critical point to get the larger dynamic range, for example. So that's so far speculation. The analytical computations show that there is this dichotomy, that if we have a possibility for noise or short observation window, we might want to move away from critical point. So we can add that this available processing time potentially controls the optimal time scale, at least if we look at optimality in a sense of finding the largest dynamical range, the largest number of signals we could discriminate, largest ways of reaction. So that's it. These are people I'm happy to work with and collaborators on theory and in experiments, and I'm happy to take any questions.